Okay, so as usual, welcome everyone. So we're gonna start in 5 minutes so that people can join in and then we can start our today's class. So, yeah, I'll brief some things up once people are in the meeting. Okay. I guess we can start now, right? So there's good enough number of you guys available here. So, yeah, few disclaimers as well as few questions. Disclaimer first, as yeah, you already know the meeting will automatically end after 1 hour. So I request you all to rejoin immediately because I will not be leaving the meeting with the same link. And again, I apologize for this. And this was the disclaimer. The question is, how many of you are following the lectures as they are released? Like, for example, if week 3 lectures are out, so are you up to date with it? Or whatever we have discussed, were you able to get something out of these sessions? So either you can put it in the chat box, or you can unmute yourself and say. I'll also tell you why I want to know this, because imagine if I keep uh, giving you lectures but they are not beneficial to you, they are not helping you, then either I have to change how you guys understand things, or I have to slow down, or I have to move fast. So all those things needs to be taken care of. So any one of you can just drop things in the chat box. That are you guys able to follow the lectures which are there on NPTEL website? Second, do you have any questions from the previous sessions which we had, and you want some clarification, or you want to go on some other topic? That is what I want to know, because things from now onwards, they'll become little bit more complex. So if you're not keeping up with the classes, they'll just make things more confusing for you guys, and I want you to understand everything from the scratch, like whatever you know about immunology from your previous exposure. Keeping that aside, we can start from the scratch as well. Okay, this will help you to have a better understanding of the subject. So as we move along the third week and the coming weeks as well, things will go into complexity. Till now it was just the brief overview of immunology, then the components of immunology, then who are the effectors and the participants in the immune system. Now we are going at the molecular level of things. Okay, so if you have confusions, or you are having facing some difficulty in understanding them, then the molecular concepts will eventually become more I won't say difficult, but definitely more challenging for you to grasp the importance of that. So yeah, please, just anyone can just tell me whether they are up to date with the lectures or not, or just raise your hand if you are up to date with the lectures. Okay, so none of you have followed the sessions. Okay, then discussing questions will be little difficult. Okay, keeping that aside, so what we'll do is, wherever we ended last time, we're gonna move ahead from that, and we are not particularly focusing on what was discussed in this week. We will eventually come towards it, but this was not the part of this week. This is part of the last week, like week second classes. Okay, so just giving a brief revision. Till now we understood there are innate immunity and adaptive immunity. In adaptive immunity, we have two components, the humoral component and the cellular component. Okay. We also understood the response of our body, how it reacts to when the antigen enters first time versus when it enters the second time, and how our system basically does that. We also okay, remind me whether we have discussed the inflammation pathway or not, because that was a crucial part of. Okay, we have not discussed what's here. Okay, so I'll start from here. Okay, so what it says, this is just the basic specificity of receptors for innate immunity or the innate response versus the adaptive immunity. So why do we need receptors in the first place? Okay, so this question should arise. So the basic reason why we have receptors is because we need communication. So imagine if something is happening, take your body. Okay, you are sitting at your place, some might be in AC room, some might be in cooler rooms, or some might be just using a ceiling fan. Okay, all of these three different types of environments, you are perceiving it differently, right? If you sit in an AC room, you feel cool, but it's not the same cool you feel when there is only ceiling fan, right? So how you can distinguish both of these? At the end of the day, both of them are wind, right? Air, that's it. So we can communicate, we can understand what is going around us from our sensory organs, right? From our eyes, from our ears, from our skin. Okay, all these are our sensory organs. Now imagine you are a very small cell sitting somewhere in your body who has zero contact from the outside world. Okay, but we call cell as the fundamental unit of life, correct? Which means anything which is changing outside, we have to get adapted for that change, and the adaptation comes from the organism. The organism is made up of organ system. Organ systems are made up of organs, and finally organs are made up of tissue, and tissue is made up of a cell. Okay, so if we want to adapt or buffer any change which is there in the atmosphere, there in the surrounding, our cells need to know that, so that they can change something in them which can be depicted at the organism's level, right? So when organism is changing or adapting, the cell has already made that decision that something needs to change so that this organism can change and adapt. But how does the cell knows that there is something going on? It is the responsibility of the cell, and to make sure that they are able to understand, get adapted, they have receptors on their cells. Okay, there are different types of receptors, there are different types of proteins, there are different types of transporters, channels. All these things are there on the cell membrane to make sure that the cell can communicate from the outside world in a very brief and overview way. Okay, so when we are talking with respect to the immune system, or when we are talking about only the cells which are involved in the immune system, so there were two major branches, the innate immunity and the adaptive immunity. Now we are talking about the receptors which are involved in the cells playing role in the immunity. Okay, so for innate immunity, the feature of innate immunity is specificity. Okay, if we talk about specificity, by now you guys are well aware and you can easily answer this question as well, whether innate immunity or adaptive immunity, which immune response is more specific, right? Just give it a try and type in the chat box, according to you, which okay, I'm sorry Arnab, I just saw your text, and you can unmute yourself, right? Yeah.
Oh, yeah.
Yeah.
Am I audible?
Yeah, you are.
Yeah. So actually, I just had one doubt from the week 2 class, basically. So when we are speaking about innate to adaptive transition, I understand that adaptive, I mean, if I want to check whether my response is a part of innate or adaptive, so this is my objective. And to speak about adaptive, I know there will be antibodies, there will be some clinical symptoms, hypersensitive reactions. But is there any kind of upregulation or downregulation of cellular marker or release of any particular cytokines that we can identify as a like marker or as a barrier to identify that my response is transitioning from innate to the adaptive phase?
Okay, so first thing first, what we need to have a very clear vision of is, it's not a clear cut transition. Okay, it's not that at 0.1 second you are in a innate phase and then at 1 second you are in the adaptive phase. Okay, so it's a gradual transition via the help of effector molecules such as cytokines and interleukins. Okay, so primary marker of innate response is the inflammatory response. Okay, once the inflammation is going on, few of the cytokines, for example, if I remember correctly, IL-6 is one of such interleukin, which is an inflammatory cytokine. This cytokine is also involved in letting the adaptive arm of the immune system know that there is inflammation going on. So IL-6 doesn't have only one work. For example, to induce the inflammation. It is a cytokine, it's a small molecule, right? It can have other functions as well. So once due to the inflammation, some cytokine is released, that cytokine will also let the adaptive arm know that, see, at this place something is going on, you need to rush here. And the place will be your secondary lymphoid organs, right? So it's not a clear-cut distinction between innate and adaptive. So and also this is how we learn. So during the initial classes, this is what I mentioned, that how we are studying and trying to understand the subject has nothing to do with how it actually works. Okay, right now I'm talking to you, so you are hearing, you are listening, you are understanding, you are processing, and you are giving response as well. All these things are happening simultaneously. It's not like that you are listening first, then you are understanding, then you are thinking, then you are responding, right? So all these things happen simultaneously. Same thing goes with anything which is in your going inside your system. So I guess in the second slide itself we have inflammation responses discussion. So if you remember your week second sessions, you will know the effector molecules, right? So we studied about the innate effector molecules as well as the adaptive effector molecules. So innate effector molecules were actually the effector molecules which are letting the adaptive system know that there is something. Now even the innate immune response can have different pathways, correct? What do I mean by different pathways? So imagine a mast cell was there, an infection came, mast cell was there, it phagocytized, and it said, no, we don't need adaptive immune response here because we have already controlled it, right? So now we don't need any connection between innate and adaptive. Now imagine there is a parasite. Okay, a parasite is an extracellular antigen. So now the innate response will try to control it, but the parasite is very large, very big, so they'll fail to control it. So what they'll do here, n number of things can happen. The first thing will be the release of heparin from the mast cells, right? But even the release of heparin is controlled by, or say supported by, the adaptive immune system, T cells, cytotoxic T cells, all those things will come into play. So which pathway innate immune system is taking, that also determines how it will connect the adaptive immune response. So there you can pinpoint some cytokines that, oh, it's playing role here as well as in the adaptive, but it will be a little bit difficult to clearly categorize that, oh, now because of this effector molecule we are into adaptive phase or we are in innate phase. It's like a continuous dynamic process. I hope I'm able to little bit clarify your doubt.
Yeah, sure, definitely.
Yeah.
I mean, I have better understanding now.
Okay.
Thank you.
Thank you so much. So what I was saying here, when we are talking about specificity, I want you guys to text me, according to your basal understanding, which immune response. So our question was regarding specificity. So I want you guys to text me, according to your knowledge and whatever you have understood till now, which arm of immune response is more specific or has more specificity. Is it innate immune response or adaptive immune response? And please, I want your involvement in the class, because it won't help you if you just come here and listen me speaking, right? Okay, cool. Few other people, like some of you can just type, right? It's just a basic chat. Okay, so yes, the adaptive immune response will be more specific, right? So when we talk about innate immunity specificity, so structures shared by class of microbes, that is PAMs, they are different microbes. Irrespective of microbe, the innate immune response will recognize them. Hence, the specificity is very less. Once the adaptive immune system comes into play, adaptive immune system response will have personalized antibody or personalized response for every single antigen or every single microbe, making it more specific for the infection. Okay, when we talk about microbial molecules recognized, so about 1000 molecular patterns, that is PAMs, essential for survival of microbes, usually cannot. Okay, so these are basic information. A question should arise in your head that when we are saying, okay, we have cells, we have receptors, and they are recognizing whatever antigen comes, right? So imagine we are exposed to thousands and thousands and thousands of microbes on daily basis, right? So do we have only one type of cell receptor which will recognize all those 1000 microbes or thousands and thousands of microbes? No, right? So what we are identifying or what coat the microbe is having on it, which can be recognized by our system, that oh, this coat we have not seen in our body, which means this is an outsider's coat. Okay, so microbes are very smart. What they can do is they can change their coat now. So if they change their attire completely, our system won't be able to recognize them. But why microbes are not doing that? Because those things are important for the microbe survival, basically. That is why they also cannot change it, and this property our system exploits. Okay, when we talk about adaptive immunity, so more than 10^7 antigens we are able to recognize in our cells during our, and it is again, it's a very continuous process. So whatever your repertoire of antibodies are there, say 1 week before, they are different after 1 week. Okay, because it's a continuous process, continuous adaptation process. When we talk about receptors, so in innate immunity, they are located in plasma membrane, endosomal membrane, and in cytosol. So all these things are part of cell itself. So they are present in the surface of the cell, as well as in the endosomes, right, and as well as in the cytosol of the cell. For receptors of adaptive immunity, because cell adaptive immunity is only getting stimulated when the innate immunity is telling them, and innate immunity can only tell them when they have processed the antigen. Hence, the adaptive immunity doesn't need to have receptors inside the cell, because they are recognizing the antigen which is presented by the APCs or the antigen presenting cells to the adaptive immunity. Okay, numbers and types of receptors. So there are around, for innate immunity, innate immune response, a microbe can be dealt with hundreds different types of receptors, and these receptors are distributed on the surface as well as in the cytosol. Okay, for adaptive immunity, there are only two types of receptors. Okay, Ig, that is an antibody, and the T cell receptor. But these two types of receptors have hundreds and hundreds of variations, which give the adaptive immune response its specificity. Okay, then distribution of receptors. So non-clonal, that is identical receptors on all cells that express the receptor, and here clonal, clones of lymphocytes with distinct specificities express different receptors. This we'll discuss in the coming slide, because this the clonal selection part was discussed in this week. Okay, genes encoding receptors. So basically because it is innate immune response, so basically in the germline, whatever you got, you have that. Okay, but adaptive immunity, as mentioned earlier, it's a continuous process, continuous adaptive process. So you need a lot of recombinations to make sure that you have millions of variations in cells to have high specificity. Can you see how I'm connecting all these features, right? So everything is related. Then discrimination of self and non-self. So in innate immunity and adaptive immunity, obviously because it's an immune response, so it should have, and they actually have a clear distinguishment between what is their own and what is from the outside. So healthy host cells are not recognized, or they may express molecules that prevent innate immune reactions. Okay, yes, based on elimination or inactivation of self-reactive lymphocytes may be imperfect. And why we are saying imperfect? Because we know the cases of autoimmunity exist. So what is basically autoimmune disorders? Your own adaptive immune system starts making antibodies for your own cells, and they are just killing them without knowing whether they are part of the host or they are from the antigen. Okay, for inflammatory response, so this is the primary reaction of our body whenever an infection comes inside us. Okay, and this is a marker that our immune response has taken place. Which type of immune response? It's primarily innate immune response, but obviously it can have adaptive counterpart as well. Okay, so for the second week this thing was discussed very in deep, so here I'm just gonna give you the overview of whatever was discussed there. Okay, so firstly, imagine this is your skin. You got some rash, you got some wound or injury. So once your skin is open, your primary physiological barrier is broken. Okay, now microbes have direct entry for going inside. The skin is not there anymore to protect your body from the outside infections. Okay, so the infections which are constantly there will finally come. Now just below there, and it can happen, so imagine if this is, we are talking about your hands or arms. Okay, so just below the top layer of your skin there are loads and loads of cells which are residing there. And what is the role of those cells? So those cells won't come when there is barrier breakage. There are other types of cells, but these cells are constantly there and they're searching for something. That is their whole job, that is their whole purpose. Okay, now imagine the microbes enters, then what happens? These cells which are there, they'll keep searching for things, and the moment they find something that, oh, something new has entered, they'll secrete some cytokines or some sort of signaling molecules. Okay, and by some kind of molecules I mean cytokines basically. So once they secrete cytokines, then what happens is it leads to the permeation of cells which are there flowing in your blood. Okay, so our bloods are made up of cells. Cells are joined together, but they are flexible as well, right? So if you remember, there were junctions between cells, right? So tight junction, gap junction, all those things were there when we study about physiology and how tissues are formed. Correct. So imagine this is a continuous layer, but still there are connections between them, right? So in normal conditions these cells are not going in between the tissue or the injured space, but once some molecules or chemical entities are released, what they make is they make these junctions loose. Once these junctions are loose, these cells which are there in your blood, they can easily come out into the injured space. Correct. Now depending upon the microbe or the infection, the necessary steps will be taken. Okay, the first step will be to engulf the microbe which has entered and present it to the adaptive immune system. So complement we have not yet discussed, hence I am not saying too much about it. So we'll have macrophages which will eat this microbe, send it to the adaptive immune system, and say ki, see, this is the type of microbe we got, then what it will do, it will release antibodies which will again come at this place and gets attached to the microbes, because there are n number of microbes, right? Not only one microbe entered, multiple microbes are there. So it will attach, the antibodies will attach themselves to the microbe. Now again, depending upon what we want from this, whether we want the complement system to be activated, whether we want this cell to be directly killed here itself, or we want this cell first to be engulfed and then get killed. You remember there are n number of types of T helper cells, cytotoxic T cells. So if we decide that this microbes is supposed to be phagocytized and then killed, so we need T helper cells here, not cytotoxic T cells. So once the antibody comes, then molecules, or say again chemokines or cytokines, these cells will also come here and engulf these n number of pathogens which are there, and then at the end of the day, with the help of high acidic nature of lysosomes or endosomes, the microbe will be dealt with. This is a very brief overview of how the inflammation works. And why we have inflammation? Because our blood vessels are leaky now. So the blood and heat, because there are a lot of small molecules which are being released here and there, that particular area becomes red, swollen, because all these cells are coming in to the surface, right? It becomes red, swollen, and little bit warmer. Okay, so this is the very brief overview of how the inflammation works. Okay, this is again the little bit more detailed steps for inflammation, how the inflammation works. I'm not going to discuss this particular thing here unless you have seen the second week's lecture, because similar thing has been discussed there as well. So you guys have a good look of this. You see all the molecules which you are seeing here, and you can categorize them what they are. For example, if I say selectin, okay, you should know what a selectin molecule is. Okay, and basic steps in the inflammation is, because here we said cell will come here, right, at the site of infection, but how will the cell exactly come here? So there are n number of steps which can be seen once the inflammation is taking place. Okay, so the first thing the cell which has to do to migrate towards the infection is rolling mechanism. Then integrins activated by the chemokines, so these are the markers of inflammation. Then adhesion at the surface and where it is required, and then finally migration from the blood vessels towards the infection site. So remember selectins, then there were integrins, all these are adhesion molecules. So once again, if we talk about on what is the major purpose of these, it's that they need to get information, they are receptors at the end of the day. Okay, so once selectin is there, it is bound, then the cell knows, oh, I have to do something else. If integrin is there, if integrin is bound, then the cell knows, oh, there is something else which I have to do. Okay, so the cell will come, it will roll, then the integrins will activate the chemokines. These chemokines will further invite more cells. At the end of the day, they'll reach the infection site. For your basic understanding, you should not worry too much about that, oh, what is the first step, second step, third step. No, it's a very intuitive thing, right? Imagine someone has called you from somewhere that, oh, come, let's go, we'll have a cup of tea. Okay, so what will be the first step? First step will be that you got that call. So that call is a sign, it's a signal that you got. Then the next step will be you'll change your place, you'll change your location, right? Then the third step will be you'll move towards the destination wherever it is decided, right? And the fourth step will be you have finally reached the destination, then you will do your action. It's very intuitive. Similar thing you can fit here as well. This cell got some signal and started rolling, which was already rolling. They were doing their own business, but they now got a signal. that signal got another signal which made their motion little bit more directed. Okay, then once the direction is there, so that they don't flow here and there, and they have a location now, so they have a adhesion where they are supposed to go towards the wounded side, and then at the end of the day they'll finally reach their destination. Okay, so these things, instead of taking them by their name, they are little bit intuitive and try to understand rather than mug up. Okay, now again, okay, so these are the types of receptors which are there, and they are part of innate immune response. So in the previous slides we have seen that the receptors of innate immune system, they are present on the surface as well as inside the cell, right? And they are the ones which are recognizing the microbes from their PAMs, and these receptors are called PRRs. Okay, so what are these receptors? So the surface receptor, if we start from top here, the surface receptor of immune response, or PRR, first one is TLR, called toll-like receptor, which recognizes the bacterial cell wall lipids. So if a bacteria is here, it will have a cell wall that will be recognized by the TLR type of receptors. Okay, another surface receptor, C-type lectin receptor, it recognizes the microbial polysaccharide. So the sugars which are there on the microbe will be recognized by the C-type lectin receptors. Now imagine these two failed and microbes somehow entered inside the cell. So here there are receptors which will recognize the entered microbe. So first, if we talk about the cytosolic type of innate immune receptors, they are Nod-like receptors, or called NLR. So what they recognize? They recognizes bacterial cell wall lipids, products of damaged host cells, which means imagine a bacteria was able to enter inside the cell. So they might have damaged the cell wall at some point, right? So NLR, or Nod-like receptor, will identify that. Another cytosolic receptor is Rig-like receptor, or RLR. So what this receptor recognizes? It recognizes the viral RNA, because remember, all the microbes, they do not completely enter. A well-known mechanism for the viral infection is that the viral will sit at the membrane and will just send its nucleic acid here, right? So there will be no cell wall, there will be no lipids, there will be no polysaccharides to be identified by these receptors, correct? So those type of infections will be recognized by the RLR, or the Rig-like receptors. Another type of cytosolic receptor is cytosolic DNA sensor, or CDS. It basically recognizes the microbial DNA. So this one is for the RNA and this one is for the DNA. Okay, now imagine even every single thing of this is not working, or failed. Not working, I won't say, failed for some reasons. So the microbe who is seeking some some help, because all these receptors are there in the cytosol, so the microbe somehow escaped them and entered into an endosome. Okay, so in an endosome, what you can have? You have another toll-like receptor which recognizes the nucleic acid of the ingested microbe. What do we mean by ingested microbe? Because a microbe was sitting here, right? It was phagocytized by the membrane, by the cell, and after after the phagocytosis, this endosome was formed. Now the microbe is in this endosome. Due to the change in the pH of this endosome, right, what will happen? The microbe will be killed and its DNA, or the nucleic acid, will be in this endosome. That thing will be identified by another TLR, or toll-like receptor. So a very basic question which generally comes in the examinations is, do we have TLRs inside the cell? So the answer will be yes. We are not talking here about types of TLRs. So there is TLR 1, 2, 1, 2, 2, 4, 3, 6. Okay, all these TLRs are there, but we are just saying a TLR is present on the plasma membrane as well as in the inside the cell. Why we can remember this? Because this TLR is present in the endosomal membrane, and from where this endosome is formed? It is formed from the plasma membrane itself. Okay, so again this is pretty intuitive. Cool. Moving forward, so this is how we'll understand the activation of macrophages. So as I have told you, once the microbe or the infection happens, macrophages are already there. They are just like a little hungry monsters. Okay, and if you are a fan of say Harry Potter, you know Dementors, right? So Dementors are just floating here and there. They'll feed on anything which is moving, which is living, and which has some soul. Okay, take macrophages like that. Macrophages are constantly scanning your system here and there. Okay, but they'll get activated only when they find something which doesn't belong to the host. Okay, so this is your plasma membrane and this is your cell. Now a microbe entered, and suppose we are talking about TLR, toll-like receptor. So this identifies the cell wall lipids, right? So this is a microbe, and this receptor is identifying the lipids of this bacteria. So once it recognizes, something happens in the downstream or inside the cell receptor. Okay, it will get activated. This receptor will get activated. Once TLR is activated, what does activation means? This activation means it will now have a downstream cascading of events. And how do we do events inside a cell? By small molecular entities which are called cytokines. Okay, so once this TLR recognizes a microbial cell wall lipid, it will activate, and after activation it will release some cytokines. Or what type of cytokines? TNF. Okay, TNF is tumor necrosis factor. These are just molecules, just take that. Okay, so they'll release TNF, IL-1, IL-6, IL-12. So once these small molecules are released, this is a signal for the cell to do what? To do inflammation, to let the adaptive immunity know that a microbe has been recognized, has been identified, and we need your help. Okay, remember there was another kind of receptor on the cell, cytokine receptor. So cytokines such as interferon gamma is released. So INF gamma will be released. Imagine there is another cell here. Let me use my bad drawing skills. Imagine another cell is here, exactly like this. Okay, so this microbe was present here, so it got it activated the TLR, and then we got the inflammation response. Now imagine there is adjacent cell, and in this a viral infection has taken place. Okay, so this cell is virus infected now. So this cell is dealing with all the things. So you remember we have Rig-like factors like this. So these Rig-like receptors are working in this cell, but in the meantime this cell should also tell their adjacent cells that a virus has entered, so you should be prepared. Okay, so this infected cell, how will it communicate to its adjacent cell? By releasing cytokines or small molecules. One such small molecule is called interferon gamma. Okay, so once interferon gamma is released, now imagine if this cell doesn't have a receptor for that. Even if this cell is releasing interferon gamma, this cell won't even know, because they don't have the sensor, they don't have the receptor. So one such receptor, which is called cytokine receptors, it identifies, suppose, interferon gamma. Once this cell knows that, oh, interferon gamma has come, what it will do? It will go phagocyte oxidase. So it's basically change the nature of the oxidation states or the availability of ions inside the cell. It will lead to the killing of the microbe, because if this cell is infected, the chances are this cell might also get the infection soon. So once this will get the infection, the cell will already be prepared, and it will kill the microbe. Another type of receptors are the complement system. So all these systems basically do the killing of the microbe. How they are killing the microbe, the route might be different. Even the inflammation helps in killing or controlling the infection. So how they are doing, the path which they are choosing, it completely depends upon which receptor is being activated. Okay, so innate immune response, which we said, oh, it's very non-specific and all that. It's non-specific because in the downstream of these receptors, the ultimate goal is to kill the microbe, and this can be decided by the pathway which is activated, and these pathways activation depends upon the type of receptor which is being activated, and the type of receptor will be activated once they recognize the PAMs. There are different types of PAMs. Here we have discussed only few, such as bacterial cell wall, such as lectin molecule or polysaccharide, such as viral DNA, such as viral RNA, such or normal DNA. These all will be PAMs. Okay, so it's a very intricate business of how our body fights with the infection. Okay, so from here onwards we gonna enter into a new territory. So during the beginning of this particular class, I told you that till here we were just having how the system works, what are the components, what are the molecules. We are just giving you the brief introduction of them. Oh, this is how the system will work. But now we'll delve into the molecular component of immunity. So imagine when I am saying INOS or nitric oxide, it's a small molecule. How it is affecting the cell, how it is killing the cell, what is happening at the molecular level. Now we're gonna understand that part. So it will be a completely different experience of immunology. Till now we were just seeing how the participants are playing role. Now we'll see what is making that role to happen. Okay, so I hope I am not moving too fast. If I am too fast, please let me know, and if I am going okay, then please also tell that, because you guys are not giving me response, so I don't know. Is it okay, not okay? Then we'll discuss questions in the second half as well. You can unmute and say, or you can just say in the chat box. Okay, fine. So I got few yes, that the speed is okay, things are fine. So now we'll enter into the new territory of immunity or the immune response. It's same. So any system, irrespective of what you are studying, I mean, okay, any system can be understood from different perspectives, correct? So imagine if I want to understand a cell can be understood in the light of biology, which is the dedicated field which study the living system, right? But a cell is made up of lipids, a membrane is made up of lipids, proteins, all these components are there, right? So which means it's a material at the end of the day, right? So a lipid, if you can make something out of it, it's a material. So a cell which is living, and we are reading with respect to it, we are understanding cell from biological aspect. The same cell can be understood from physical aspect as well, because it's material, right? So we can study about what is the tensile strength of the cell membrane, or what is the Young's modulus of the cell, or how much shear this cell can take, or what are the physical properties this cell is posing, what is the mechanical strength of this cell, right? All these things can also be studied, but the system is same. Here the system is cell. Now the same cell which we try to understand via biological means, we also understood via physical means. Now the same system can also be understood from chemical aspect as well, right? So imagine, okay, if I am saying the membrane is made up of lipids, so what are lipids, what type of molecules are lipids, what are proteins, what kind of chemical entities are proteins, what are the groups associated with it, is it carbon, is it hydrogen, is it phosphate, oxygen, what? So this is the chemical aspect of the same cell, right? So now where we are going is, we're gonna try to understand immune system from the point of view of molecules, or from the gene level, or the protein level, or the transcription level. Okay, all these things, the molecular, the macro term will be the molecular aspects. So antigen recognition in the adaptive immune system, structure of lymphocyte antigen receptors, and development of immune repertoires. B and T lymphocytes expresses different receptors that recognize antigens. They can be membrane bound antibodies on B cell and T cell receptors on T lymphocytes. Till here it's very clear what we are trying to do. What we are trying to do is, a microbe has entered. Till now we said, oh, a TLR will recognize lipid cell wall of a bacteria, a lectin receptor will recognize the polysaccharide. But how they are recognizing it, what is the major player which is helping the receptor of our cell to recognize the polysaccharide of the microbe? Okay, this we'll see. So this process is basically called antigen recognition. So how our system is identifying the antigen. So some questions before going into this topic, we should have it in mind, and this will help us understand the system better. So the first question is, how do the antigen receptors of lymphocytes recognize extremely diverse antigens and transmit activating signals to the cells, right? Second is, what are the differences in the recognition properties of antigen receptors on B cells and T cells? Third, how is the vast diversity of receptors in the lymphocyte repertoire generated? The diversity of antigen recognition implies the existence of many structurally different antigen receptor proteins, more than can be encoded in the inherited genome. Therefore, a special mechanism must exist for generating this diversity. Why this question is so pertinent? Because if you remember here, we said more than 10 raised to power 7 antigens we can recognize at a given point, and this can change obviously. So this 10 raised to power 7 ability of our system to recognize these many different types of microbes, how we are attaining this? Because we have limited genome, limited things are transcribed, proteins are there. So how come 10 raised to power 7 variability we can recognize? So these are few persistent questions which we'll try to answer in the coming slides and the coming lectures as well. Okay, so first we'll talk about the antigen receptors of lymphocytes. So till now, in the last we studied about the innate immunities receptor, like the cells which play a role in the innate immune response, what sort of receptors they have. Now we are into adaptive arm. Now we are saying, in the adaptive immunity, what sort of receptors are present. Okay, you all have heard the name, but now, as I have said, we'll go into little bit more deeper and molecular basis of those things. Okay, So when we talk about antigen receptors, we have two major types of antigen receptors. First is the antibody, second is the T cell receptor. So one clear distinguishment you can see, the feature which distinguishes these two types of antigens are membrane form, which means whether this receptor is membrane associated or whether it is not membrane associated. So you need to link this with adaptive immunity when we said that there are two different types of adaptive immunity. Remember, humoral immunity and cell mediated immunity. So in humoral immunity, things were secreted which were taking part in the adaptive immunity, and in cell mediated ones nothing was secreted, correct? So that thing can be linked here as well. So when we talk about antibody can be membrane bound as well as can be secretory in nature, while T cell receptor is membrane bound only. Okay, then the types of antigens which are recognized by antibody is macromolecules such as proteins, polysaccharides, lipids, nucleic acids and small chemicals as well. Conformational and linear epitopes, which means 3D protein structures, as well as if the protein is processed and it is in the primary form. For T cell receptors, it recognizes mainly peptide or polypeptides or the processed antigen, which is displayed by the MHC molecules on APCs. So they only recognizes linear epitopes, or in one word we can say processed antigens is recognized by T cell receptors. What is the diversity of antibody and T cell receptor? So antibodies, each clone has a unique specificity, potentially for 10⁻11 distinct specificities. So remember the question which is pertinent, right? How is this high specificity achieved by our system? So you can see it is too much, 10⁻11 is too high, right? For T cell receptors, each clone has a unique specificity, potential for 10⁻16 distinct specificities, right? Antigen recognition is mediated by variable region of heavy and light chain of membrane immunoglobulin. So these are new terms. If you guys have not gone through this particular week's lecture, then it will be difficult, but I guess I think today I might not be able to cover these topics. We might cover this in the next class. Okay. And in T cell receptor, the variable region of alpha beta, so you can just skip this if you have not gone through this week's lecture, but if you have gone through this week's lecture, you know what a variable region is and what are the alpha and beta chains. Okay, so antigen binding site in antibody is made up of 3 CDRs, or these are basically variable regions in the antibody. Okay, so as you can see, this is antibody, right? So this region of antibody is made up of CDRs. So this CDRs,
इथं काय करायचंय आणखी ही लेन्स लावते की पुढच्या हाताला लावणार आहे आधी दोन्ही लावते ते 2 पंख लावून घेते फक्त हाताला थोडं लावायचे की पहिला.
Can you guys please check your mic once? Can you guys please check your mic once and keep yourself muted if you don't have to ask any question. So we'll give 1 minute so that people can join in, and till now you can just go through this table and this is what we're gonna study. So CDRs, if you know these terminologies, so CDRs are basically complementarity determining regions, MHCs are major histocompatibility complexes, APCs, these terms should be very okay to you by now. Okay, they should not sound like some completely new terms. So we'll give just 1 minute, okay, so that people can just come and nothing can be like no loss is there for them. I think we can proceed now. So yeah, we were discussing about the antigen recognition, and now we are talking about antigen binding site. So you know, these are things which are associated with the structure of the antibody as well as the structure of T cell receptor. Now the affinity of antigen binding, you guys already know the term affinity, right? So it has nothing new to do with respect to the immune system. A basic affinity means the strength, how good you are in making the interaction. Okay, so this is the general and layman's way of defining affinity. So for antibody, the affinity ranges from 10 raised to power minus 7 to 10 to power minus m molar. Average affinity of Igs increases during immune response to protein antigen. The dissociation constant for TCR or T cell receptor is 10 to the power minus 5 to 10 to the power minus 7. No change during immune response because they are fixed, right? They are in cell. They are present on the membrane. So nothing can now change in this particular cell. Okay, so the affinity for TCRs is stable and it doesn't change with respect to the immune response. Signaling functions which are mediated by antibodies are proteins associated with membranes and proteins associated with the TCR. So this is signaling. When the signaling will happen, again some common things are here. A signaling will happen. We have seen a very brief view of signaling or activation here. So some in receptor something came and bind, and then the activation happened and the signaling lead to the your action at the end of the day, right? So signaling can happen when mostly like most of the time when the receptor is associated with the membrane itself. Okay, so in case of antibodies which are membrane associated can provide the signaling or the activation of the cell. For TCRs, as all the receptors are always membrane bound, they'll have other supporters for that activation as well. When we talk about effector functions, so it can be again this is structural things. I think it's best if we can directly go to the structure of antigen and the T cell receptor. So we'll see few basic overviews of B cell receptor first. So B cell receptors are membrane bound antibodies. Why we are saying membrane bound antibodies? Because these receptors, once they don't have the transmembrane domain, they get secretory, and the secreted form of B cell receptor is called antibody. Okay, so B cell receptors are membrane bound antibodies. They recognize diverse chemical structures such as proteins, lipids, carbohydrates, nucleic acids, and other small entities. They detect native shapes, conformations of macromolecules. Activated B cells secrete antibodies with identical specificity. Functions of B cell receptor is bind or eliminate microbes and toxins in humoral immunity. So these are pretty self-explanatory points, because all this we have discussed in the previous classes. Okay. When we talk about T cell receptor, like what are the major differences between B cell receptor and T cell receptor? So T cell receptor, they recognize only peptides bound to major histocompatibility complex molecules. Antigens are from intracellular proteins or extracellular proteins processed by APCs or antigen presenting cells. They do not bind, that is T cell receptor do not bind to free or soluble antigens. What are the functions of T cell receptors? Some of the functions. So they target the infected cells. So this will be done by cytotoxic T cells, or tumors, or antigen presenting cells, that is Th cells, T helper cells, right? So the key differences between B cell receptor and T cell receptor is B cell receptor broad molecular recognition in native form, okay, because they can recognize all these macromolecules, right? In T cell receptor, they can only recognize antigen when it is being presented to them. So if B cells are not there or APCs are not there, T cell won't even know that there is infection. Okay, so how that knowing is going on? That knowing is going on by the antigen presenting cell. When they have the MHC molecule, they are presenting the antigen, then only the TCR is able to recognize this. If this antigen was free here and there, the TCR won't able to recognize that. But a B cell receptor can recognize the antigen, as you can see here. Okay. What are the structures of structures of antigen receptor molecules? So B cell receptor structure and T cell structure receptor. So the domain organizations. So in domains of B cell receptor and T cell receptor, you have variable region and constant region in both of them. Okay, so in variable region, the antigen recognition differ between lymphocyte clones, which means variable regions are different in different lymphocytic cells. So cell A and cell B, though both of them are B lymphocyte or B cells, but both of them will have different ability to identify the antigen. Constant region, so the structural integrity in binding to signaling molecules conserved across clones, which means if a B cell receptor is there on A cell and B cell, A cell can recognize one type of microbe, B cell can recognize another type of microbe. So their variable region is different, but in B cell 1 and B cell 2, constant regions are same. So that hence the name constant. Okay. The sequence variation, so these receptors are protein in nature, right? So proteins have amino acids. So what is the sequence and the conservation of these amino acids between the receptors? So variation concentrated in hypervariable regions or complementarity determining regions. CDRs, that is complementarity determining regions, complementary to the antigen shape, leads to the specificity. So here we are talking about the key structural features. What we are saying is that this is the constant region of an antibody. It is same between cell A and cell B, but this is the variable region. This variable region is different in cell A and cell B. This variable region, which is recognizing the antigen at the end of the day with respect to the sequence, because there are also amino acids here in this one, so the sequence variability is more in this region in comparison to this region. Okay, then the functional design. So variation limited to small regions, that is maximizes antigen binding diversity while maintaining receptor structure and genetic basis. Special mechanisms in developing lymphocytes generate diverse V regions for unique antigen receptors, and we'll see this in the coming slide, that how basically the specificity and the variability within the lymphocytes is coming from. Now we talk about the signals. So the association with invariant protein. Antigen receptor polypeptide pair with invariant membrane protein. Okay, so this is little bit more text stuff. I'll give you the overall conclusion. So the overall conclusion is, we are discussing about lymphocytes. There are two different types of lymphocytes, B lymphocyte and T lymphocyte. In B lymphocyte, the cells which are B lymphocyte, they are called B cells. B cells will have B cell receptors. T lymphocytes are the cells which, like, they are called T cells. T cells will have T cell receptors. B cell receptors can be membrane bound as well as secretory. Once they are secreted, they are called antibody. So this is still B cell things we are talking about. For T cells, they are only membrane bound. Okay, this is the overall crux of all these things which we'll discuss. So to make this little bit more clear, I found this very summarizing video which might help us in understanding better. So just give me 1 minute so that I can go. Okay. Let me know whether you guys can listen to the audio.
The 4 steps in this video will cover the response of the
Is the audio available to you guys? Okay, cool.
system to viral and bacterial infection, the difference between T cells and B cells, and provide an explanation of the adaptive immune system. Number 1. There are 2 branches to the immune system. The 2 branches of the immune system can go by the names specific and nonspecific, innate and adaptive, as well as some other lesser common names. For the purposes of this video, the branches will go by the names innate and adaptive. The innate immune system is the immediate and quickest response to an infection. However, it is not very specific to any type of infection. Since the innate immune response is not very specific, it often is not enough of a response to contain the infection. This is the reason for the adaptive immune response. The remainder of this video will cover the adaptive immune response. For more information on the innate immune response, see the video on inflammation on this channel. Number 2. B cells and T cells are the fundamental cells of the adaptive immune response. The adaptive immune response consists of humoral-mediated immunity and cell-mediated immunity. Humoral-mediated immunity is the name for immunity that is provided by B cells. Cell-mediated immunity is the name for the immunity provided by T cells. Humoral is Old English, referring to the humors of the body, meaning the fluids of the body. B cells are able to provide immunity in the humors, or fluids of the body, because B cells secrete antibodies to circulate throughout the blood and other fluids of the body. If an infection has reached the inside of a cell, humoral-mediated immunity will not be effective. This is why cell-mediated immunity exists. T cells protect the body from infections that have reached the inside of cells. Thus, by providing immunity in the fluids of the body through B cells, and by providing immunity from infected cells of the body through T cells, the adaptive immune response is able to provide immunity throughout the human body. Number 3. B cells and T cells contain infections in different ways. In immunology, putting an end to an infection or bringing an infection under control is often referred to as containing an infection. While B cells and T cells are both components of the adaptive immune response, they contain infections in different ways. B cells produce and secrete antibodies. Antibodies are proteins that are able to bind to bacteria and viruses. Antibodies binding bacterial cells or viruses prevents infections from spreading. The antibodies block the bacteria from growing colonies, and in the case of viruses, the antibodies bind to the viruses and prevent the viruses from infecting cells. Any disease-causing agent, such as a virus, can be referred to as a pathogen. The process of antibodies binding a pathogen to contain the infection is known as opsonization. Opsonization by antibodies not only prevents the spread of infection, but marks the bacteria or viruses for destruction. Phagocytic cells circulating in the blood and fluid surrounding the tissues of the body can detect the presence of antibodies bound to pathogens and then engulf and destroy the opsonized bacteria or viruses. These phagocytic cells are not B cells or T cells and are part of the innate immune response. So while the process of opsonization is part of the adaptive immune response, the actual destruction of pathogens involves the innate immune response. Therefore, both branches of the immune system must work together for a complete immune response to infection. Further detail on this phagocytic process is provided in the video on this channel on inflammation. When a cell becomes infected and cell-mediated immunity is needed, T cells respond by killing the infected cell. There are numerous types of T cells. Cell-mediated immunity is largely dependent on a class of T cells known as CD8 cytotoxic T cells. The CD8 cytotoxic T cells are called cytotoxic because they are able to kill other cells. When a cell becomes infected with a virus, a CD8 cytotoxic T cell responds by killing the cell infected with the virus. Since viruses replicate inside of cells, killing the cell infected with the virus stops the ability of the virus to replicate. Additionally, while often not sufficient to rid the body of cancer, CD8 cytotoxic T cells are also able to kill cancer cells when they detect cells of the human body that have become cancerous. Thus, while CD8 cytotoxic T cells are killing cells of the human body, whether virus-infected or cancerous cells, this can actually preserve health overall in the body because the killing of the infected cells can stop the spread of the virus or cancer. Cell particles and viral proteins that are released into the fluids of the body after the infected cell is killed can then be destroyed by phagocytic cells, just as is the case with opsonization. To summarize adaptive immunity, B cells respond by releasing antibodies for opsonization of pathogens, resulting in the destruction of the pathogens. T cells respond to infected cells by killing the infected cells. Specificity is the basis of adaptive immunity. As previously mentioned, adaptive immunity is often a more effective response at containing an infection than innate immunity. This is because adaptive immunity is specific. What this means is that adaptive immunity is focused on one specific type of pathogen. For example, if a person is infected with a strain of the flu, the antibodies that protect from that infection are specific only to that one strain of the flu. If those same antibodies that stopped the spread of this one strain of the flu were then exposed to a common cold virus or a streptococcal strain of bacteria, the antibodies would not be able to bind and would not at all be effective in containing the infection. This is due to the specificity of the antibody binding site. The portion of the antibody that binds to a bacterial cell or a virus is known as the binding site. The binding site on an antibody has a specific shape. This shape determines what an antibody is able to bind to. If an antibody comes across a bacterial cell or virus, and the shape of the binding site on the antibody is not a good fit for the bacterial cell or virus, The antibody is not able to bind and will simply continue to pass through the fluids of the body.
What do you think is the biggest health mistake people are making?
One B cell can secrete one type of antibody, meaning the antibodies will all have the same shape to their binding sites, resulting in the same specificity for one type of bacteria or virus. However, since all antibodies secreted from this B cell will have the same shape binding site, this means none of the antibodies secreted by this B cell will be effective at stopping a different kind of pathogen than the ones these antibodies are able to bind. For this reason, numerous B cells are needed for proper immunity. While one B cell may secrete antibodies with one binding site, another B cell will secrete antibodies with a different binding site, and so on, demonstrating the need for multiple B cells as part of the immune system. In the case of T cells, the T cell receptor is the specific site. Just as is the case with the binding site of antibodies, the shape of the T cell receptor on T cells is what determines which infected cells a T cell will bind to. If a T cell comes across a cell infected with a virus, and the T cell receptor is able to bind the infected cell, the T cell will become activated and release cytotoxic molecules that can kill the infected cell. The same T cell, however, may come across a cell infected with a different virus, and the T cell is unable to bind, and no cytotoxic molecules will be released. As is the case with antibodies, T cells are specific to being able to respond to only one kind of virus. There are numerous T cells, however, and an array of viral infections are able to be contained due to the variety of T cells in the human body. The specificity of the adaptive immune response allows for a specialized, intense immune response to one pathogen. However, this specificity can also be a limitation, as it only allows for a very narrow range of pathogens each antibody or T cell can respond to. Thanks for watching, and remember.
Okay, so this was the overview of how B cell, T cells are connected and how they are recognizing and processing things. So whatever we have discussed till here, it was summarized there in a different way. So by now, and I request you all to please go through week 3 lectures, so this thing will become more clear to you guys what we have learned. So till now we have understood that in the adaptive immune system, B lymphocyte and T lymphocytes are the key players. B lymphocytes receptors, once they are secreted, are called antibody. T cell receptors can only recognize antigen when it is processed, and there are different types of T cells and different types of B cells. Okay, that is the crux of everything. So in the next week's class, we'll discuss more about these receptors, how the antigen is binding, and then we'll go more deep into the structure of antibody. Okay, this was part of this week, so I thought that you guys have gone through the lectures, so I would have moved little bit faster in the previous slides, but it's okay, you no need to worry about it, we can go as slow as we want. So these were the questions which we have already discussed, so we'll discuss little bit more questions and new questions, I'll say. So the format is same. For easier questions, I'm gonna give you guys 1 minute, and for bigger questions, I'm gonna give you guys 3 to 5 minutes depending upon the difficulty level. And all of you needs to participate in this like basic giving answers and attempting the questions. It will help you as well as it will help you only majorly. Okay, so we'll start our question discussions for this week. So what the question says, the question is saying defensive barriers of innate immunity are anatomic, physiologic, phagocytic, inflammatory, or all of these. So you guys have 1 minute to answer this question because it's a pretty straightforward questions. Feel free to drop your answers in the chat box. 1 minute is about to be over. Try attempting these questions. This is a pretty straightforward question. And your time is up for this question. So the correct answer is all of these. Your time is up, and obviously the correct answer is all of these. So we know that innate immune system provides okay, so one person attempted correctly. So what you guys are saying, it's correct, but it's partially correct. So our innate immunity, as I have told you, it's all the things which a microbe has to face before it enters into our blood or cells, right? So it can be anatomic, it can be physiologic, it can be phagocytic, it can be inflammatory. These are the things which protects any microbe to further invade in our system. Okay. Second question, which of the following are cell types involved in innate immunity? All of you must answer this question. So the first question is the first option is natural killer cell, T cells, B cells, or all of the above. So you have 1 minute, though this question doesn't need 1 minute, but still, as the protocol suggests, your time starts now. The question is, what are the key players in innate immunity? Is it natural killer cell, T cell, B cell, or all of these? So your time is up, and let's see how many of you attempted this question. Okay, yes, correct. I'm so happy that you guys are attempting these questions. So the correct answer is natural killer cell, right? I have one added question on this. If the question was okay, how to put this question? Which of the following are cell types involved in adaptive immunity? What will be your answer then? Will it, and the options are same: natural killer cells, T cells, B cells, all of the above. What will be your answer at that time? All of the above. Great. This answer is for which question? When? For right now, right? Whatever I've asked, answer for that, correct? So why I'm saying this? Because if you remember, natural killer cells are the link between innate immune arm and the adaptive immune arm. They are the antigen presenting cells, right? So they connect innate immunity to adaptive immunity. So if the question says adaptive immunity, then you have to choose all of, though players will be T cell, B cell, but I just wanted you to remember that natural killer cells are the cells which are connecting both the arms. Okay. So next question for today is, which receptors help dendritic cells bind microbes and carry them to secondary lymphoid tissues? I'm going to read this question again. Which receptors present or help dendritic cells bind microbes and carry them to secondary lymphoid tissues? PRRs, TLRs, TCRs, or none of the above? Your 1 minute starts now. Basically, the question is asking which type of receptors are present in APCs or antigen presenting cells, which help them identify and bind the microbe and take them to the secondary lymphoid tissue. Your 1 minute is over. Let's see how many of you tried. CPRR A. So okay, we have a competition between TCR and PRR. So the correct answer is PRR. So I get what confused you. So TCR is basically T cell receptor. T cell receptors will be present on T cells. T cells are part of adaptive immunity. Adaptive immune cells are present in secondary lymphoid organs or tissues. Okay, so they are not identifying the receptor. And remember, T cell receptors cannot bind to the microbe directly. This is what we have discussed today, right? They can only recognize the antigen once it is being presented to them. And the cells which are presenting, one of such cell is called dendritic cells. So the dendritic cells, they have receptors. Those receptors are called pathogen recognition receptors, PRR. TLR or toll-like receptor is a type of PRR. Correct? I hope you guys understand what I am saying. If your confusion would have been between PRR and TLR, I could understand. Okay, so PRR is the umbrella term which says all the receptors which are present on APCs. One such receptor present on APC is also TLR. Okay, coming to the next question. This is just the basic explanation of this question. So pathogen recognition receptors or PRRs are a class of germ-line encoded receptor that recognizes pathogen associated molecular patterns, that is PAMs. The activation of PRRs is crucial for the initiation of innate immunity, which plays a key role in first line defense until more specific adaptive immunity is developed. PRRs differ in the signaling cascades and host responses activated by their engagement and in their tissue distribution. Currently identified PRRs families are the toll-like receptors, the C-type lectin receptor, the nucleotide binding oligomerization domain-like receptors, the retinoic acid inducible gene-like receptor, and the AIM2-like receptors. So these are PRRs apart from PRRs which we have discussed today. Remember, TLR, RIG-like receptor, NOD factors. Apart from them, these are other examples of PRRs. Okay. Coming to the next question for today. So the question says, which are the first cells to be recruited at the site of infection? Is it macrophage, neutrophils, eosinophils, or monocytes? Okay. So your 1 minute starts now. Your 1 minute is over. I really hope that you guys tried attempting this question. Let's see how many of you attempted. B, A. Okay. So we have confusion between B and A mostly. So the right answer is neutrophils, which are the first cells to be recruited at the site of infection. So remember, macrophages are always there. They are hunting. They are like ghost dementors, which I mentioned you. But as a response of macrophages encountering an antigen, all those steps, remember, rolling, attachment, then migration, that is happening for the neutrophils, the cells which were migrating. Okay, so to clear this doubt little bit more, because it's an important process in identifying how the sequence of events take place, we'll see this small animation. I think it's an animated movie. So we'll see that how the infection and the recognition of infection is basically taking place. Okay.
As a powerful army that protects it from various types of threats. These threats can come in the form of mechanical injuries, the entry of germs, or the entry of other foreign particles like dust. This personal army is called the immune system. Every day we encounter a huge number of bacteria, viruses, and other disease-causing organisms. However, we don't fall ill every other day, which is due to our immune system, an army of cells that is always roaming our body, ready to ward off any attack. The immune system can be broadly divided into two parts, innate and adaptive immunity. Innate immunity, or non-specific immunity, is the body's first natural defense to any intruder. This system doesn't care what it's killing. Its primary goal is to prevent any intruder from entering the body, and if it does enter, then the immune system neutralizes this intruder. It doesn't differentiate between one pathogen and another. The first component of this defensive system is our skin. Any organism trying to get into the body is stopped by the skin, our largest organ, which covers us. Secondly, there is the mucous lining of all our organs. The sticky, viscous fluid traps any pathogens trying to get past it. These are the two physical barriers. However, we also have chemical barriers, such as the lysozyme in the eyes or the acid in the stomach, which can kill pathogens trying to gain entry. The genitourinary tract and other places have their own normal flora or microbial community. These compete with pathogens for space and food, and therefore also act as a barrier. The next line of defense is inflammation, which is done by mast cells. These cells are constantly searching for suspicious objects in the body. When they find something, they release a signal in the form of histamine molecules. These alert the body, and blood is rushed to the problem area. This causes inflammation and also brings leukocytes, or white blood cells, which are soldiers in our body's cellular army. Once they come, all hell breaks loose. Sometimes, however, the intruder may not be a germ, but rather a harmless thing like a dust particle. The body still causes a full immune reaction to this intruder, which is how allergic reactions occur. In the fortress of our body, the leukocytes are VIPs. They have an all-access pass to the body, except, of course, to the brain and spinal cord. Our leukocytes come in many types. Those that belong to the innate system are the phagocytes. These cells can either patrol your body, like the neutrophils, or they can stay in certain places and wait for their cue. Neutrophils are the most abundant cells. They patrol the body and can therefore get to a breach site very quickly. These cellular soldiers kill the infectious cell and then die, which leads to the formation of pus. There are also the big bad wolves, or the macrophages. These cells are like hungry, ravenous monsters who simply engulf unwanted pathogens. Instead of roaming freely in our blood, they are collected in certain places. These cells can consume about 100 pathogens before they die, but they can also detect our own cells that have gone rogue, such as cancer cells, and kill them too. Beyond that, we also have the natural killer cells. These cells can efficiently detect when our own cells have gone rogue or are infected with, say, a virus. NKCs detect a protein produced by normal cells called the Major Histocompatibility Complex, or MHC. Basically, whenever a cell isn't normal, it stops producing this protein. The NKCs move around constantly, checking our cells for this type of deficiency. And when they find an abnormal cell, they simply bind to it, release chemicals, and destroy it. The last cells of our innate immune system are the dendritic cells. These are found in places that come in contact with the outside environment, such as the nose and lungs. They are the link between our innate and adaptive immune systems. They eat a pathogen and then carry information about it to our adaptive immune system cells. This information is produced and shared in the form of antigens. Antigens are the traces that pathogens leave behind. They are molecules found on the surface of pathogens that can be detected by our adaptive immune system for recognition. The dendritic cells pass on this information to our T cells. However, macrophages can also perform this function. Now, there is also the adaptive or acquired immune system. This system
You understood, right? Why neutrophils is the answer for this particular question? Because we are recruiting them. They are not already present there. We are recruiting them. Already present cell will be macrophages. Okay. Coming to the last question for today, and then we'll discuss more questions in the next class. Okay. So the question says, cell adhesion molecules or CAMs expressed on the surface of endothelial cells are known as mucins, selectins, integrins, or ICAM. If you have gone through week 2 lecture, then you can easily answer this. Your 1 minute starts now. Your 1 minute is over. Let's see how many of you tried attempting it. Okay, only 1% tried. So the correct answer is B, selectins. Okay, so to summarize this, here, oh sorry, I have showed you the answer for the next question. Okay, don't worry. So yeah, and I forgot to upload these slides and lectures. I'll upload this, so it's already
The
there.
reunification of North and South Korea is not just
So videos I've uploaded in my channel, but I forgot to give the link, so I'll do this like either today or by tomorrow for sure.
Welcome to BMH Learning. This tutorial is about cell adhesion molecules. So first thing we need to know is, what's cell adhesion? It's a process by which cells interact and attach to the neighboring cells. Now the connection can occur in two ways. The first is direct contact, where one cell makes immediate contact with the neighboring cell. This is known as cell-cell adhesion. Then we have indirect method. Cells in animal tissues also adhere indirectly to the components of the surrounding extracellular matrix. And this type of adhesion is called as cell matrix adhesion. Now the question is, what mediates the adhesion between these cells? So, cells bind to each other with the help of transmembrane proteins called cell adhesion molecules, also known as CAMs. Now let's talk about the structure of a CAM. They have an extracellular domain through which adhesive interactions occur. So, with the help of extracellular domain, the CAM binds to another CAM which is present on the other cell. Then we have the transmembrane segment of the protein which traverses the cell membrane of the cell. Finally, we have cytosol facing domain. This portion of the CAM recruits multifunctional adapter proteins. These adapters act as linkers that directly or indirectly connect CAMs to the elements of the cytoskeleton. These proteins can interact in different ways. The first method is homophilic binding, where one type of the cell adhesion molecules bind to the same type of cell adhesion molecules. They are also capable of heterophilic binding. meaning a cam on one cell will bind with the different cams on another cell. Now there are four major types of cams. The first is Ig cams or immunoglobulin superfamily of cell adhesion molecules. The second is cadherin. Then we have integrin. And the fourth one is selectin. We will discuss about each type in detail in separate videos. Now the cams perform very important functions. They are involved in inflammation. They are also responsible for tumorigenesis. They are very much helpful in the establishment of the blood-brain barrier. Another function is the lymphocyte homing. Next, we can say they are also involved in regulation of the apoptosis. Apart from these, the cams provide a means for the bidirectional transfer of information between the exterior and the interior of the cells. In other words, cell signaling. This was all. Thanks for watching.
Why do top product
Okay, so I'll free you guys for today. We'll meet in the next class, which will be again same time, Sunday. I hope by then you guys are up to date at least till week 3. I'm not expecting you to be up to date with every single week, because I understand it's a little bit heavy. But yeah, in the next week I'll expect you guys to have gone through the lectures at least till week 3. Okay, and try practicing these questions, be regular with your assignments, and yeah, enjoy your Sunday. Bye bye. Thank you. Next class will be same Sunday 4 p.m. Okay, thank you.